Hello, my name is Gabriella Gaje, and today I'm going to be talking to you about the in vivo role of 3H12-dithiol-3-thion in protection against lipopolysaccharide-induced inflammatory injury. So first things first, I want to talk to you about sepsis. And so what is it? It is a mounting public health problem with high hospital costs and ultimately leads to multiple organ failure. It mainly affects the elderly and those who are immunocompromised. It is previously defined as a combination of infection with two or more symptoms of SEERS. So here's an example of how sepsis works. It affects multiple organs at one time from one source of infection, and inflammatory responses in the body cause multiple organs to fail. So for our project, we want to simulate those conditions that are seen in sepsis in humans in mice. So we're going to be using an endotoxin called lipopolysaccharide, which is found in the walls of gram-negative bacteria. LPS, or lipopolysaccharide, has an established role in pro-inflammatory pathways. You can see from this diagram over here the mechanism by which LPS binds to the receptor and crosses the membrane and will ultimately set off the NF-kappa B pathway. The NF-kappa B pathway is what leads to the release of pro-inflammatory cytokines. We will also be using a compound called D3T, which is also 3H12-dithiol-3-thion. D3T is known for its chemoprotective properties that code for phase 2 enzymes and antioxidant genes in cancer cells. However, it is not known if the use of D3T can reduce LPS-induced oxidative and inflammatory responses. To try and test this, we are going to be using an in vivo mouse treatment. LPS is widely used to induce sepsis in animal models, so we used male mice, the C57BL6 strain, and this is the most widely used strain due to replicability and reduction in variability. We are using this model because no previous liver and kidney studies have modeled sepsis specifically with LPS and D3T. The animal treatments and sacrifices were performed at Campbell University School of Osteopathic Medicine. Once the treatments and the sacrifices were completed at Campbell, the tissues were sent over to us, and we used qRT-PCR to amplify the gene expression of the treatments. So we first isolated the RNA. Trizol reagent is used to extract RNA from the cells. The purified RNA is diluted in 10 microliters of DEPC water. RNA concentrations are measured by taking 1 microliter of the sample and measuring it in the nanodrop UV-Vis spectrophotometer. The RNA concentrations are uniform to 500 nanograms per microliter. A reverse transcriptase is used to synthesize cDNA from the 500 nanogram per microliter of RNA. cDNA reagents include DEPC treated water, 5X first strand buffer, deoxynucleotide triphosphate, or DNTP, Maloney-Murine leukemia virus reverse transcriptase, and random primers, in addition to 2 microliters of RNA. Once the cDNA is synthesized, the target genes are used to compare to the housekeeping gene of GABDH. These target genes are created using forward and reverse primers. The cDNA and target gene mixtures are placed in a clear 96-well plate prior to being synthesized. The Step 1 Plus real-time PCR system runs for 40 cycles at 95 degrees Celsius for 15 seconds, 58 degrees Celsius for 1 minute, and 60 degrees Celsius for 15 seconds. Gene expression is quantified by using comparative threshold values. So here is what the RT-PCR machine looks like, and this is what our amplification plot will look like once the cycle has been completed. So here are my results. Figure 2 shows the effects of D3T on the expression of antioxidant GCLC genes in hepatic tissue, or the liver tissue, in the LPS-treated mice. We did 4 treatments: control, LPS, D3T, and then LPS plus D3T. The relative mRNA of each above gene was evaluated by real-time PCR and data was normalized using GABDH. As you can see, there's a significant increase in GCLC gene expression with the D3T treated mice, which is the expected result, as D3T is supposed to promote antioxidant gene expression. However, when you look at this slide, we have MCP1 and IL-6 gene expression. They are both pro-inflammatory genes, and the treatment of LPS has shown significant increase in both of those gene expressions comparatively to the D3T control and LPS plus D3T. So our results show that there is significant increase in pro-inflammatory IL-6 and MCP1 genes with LPS treatment, and there's a significant reduction in the expression of IL-6 and MCP1 in the mice that are treated with LPS and D3T when compared to mice treated with only LPS, which suggests the anti-inflammatory properties of D3T. And you could also see the amplification of the antioxidant gene with the administration of D3T in the GCLC mice. These results indicate that combining LPS and D3T can reduce the inflammatory response caused by LPS, and that D3T can be used as a potential therapeutic for inflammation, specifically that is caused by the endotoxin LPS in the human body. So here are my acknowledgments. I would like to thank the UNCG Department of Biology, Campbell University School of Osteopathic Medicine, and the National Institutes of Health. Thank you for your time.